All right. So we've been talking about emotion. I mentioned the three brain regions that we're really going to be talking about are the amygdala is the main one, a little bit of hypothalamus, and the insula are the three main brain regions that we're going to talk about in this chapter that have something to do with emotion. Um, all of these are kind of lower parts of the brain. So we're not really talking that much about the frontal lobe or parts up here. Most of these are kind of in the lower half of the cortex, the ones that we're going to be talking about. The amygdala is part of the medial temporal lobe. It is adjacent to the hippocampus. And we know pretty well now that the hippocampus is involved with the formation of long-term declarative memories. We'll be talking more about the hypothalamus and insula later today, but they're all kind of these structures in the lower half of the cortex. We were talking about last time the role of the amygdala in this instructed fear paradigm. And I just want to remind you of this. This is the study where people were basically told by the researchers, you'll be seeing a square of squares of different colors. The blue square is going to shock you when you see it. It's going to cause you pain. The blue square never actually shocked anyone in this study. But they were still interested in the physiological response people had when they saw the blue square. And we did talk about this last time, so I won't spend as much time talking about this time. They are interested in the activity in the amygdala in response to that blue square and skin conductance. And they showed that both the amygdala and skin conductance, which is measured in the palms of the hands, the amount of sweat on your hands, both increased in response to the blue square compared to other color squares, and that those two responses correlated. So people that had stronger activity in the amygdala in response to the fear-inducing blue square also had stronger skin conductance responses, which suggests that there might be some link here, that the amygdala might have something to do with physiological responses that occur, physiological emotional responses that occur. So that's just a review, kind of get us back where we were last time. Moving on then, I'm trying to remember if we talked about this before. Yeah, I don't think we did. So just in case, I'll talk about this here. So they do studies on individuals with amygdala lesions compared to controls in this instructed fear paradigm. And individuals with amygdala lesions showed no increased skin conductance, which kind of further supports this idea that I was just talking about the previous slide. The amygdala, these are kind of causal data. The amygdala might be kind of causally involved with producing physiological responses during some type of emotional episode. What's interesting about the individuals with amygdala lesions is that they would see the blue square and their body would not get physiologically, aroused. They wouldn't get skin conductance increases on their hand when seeing the blue square. But if you ask these individuals whose amygdalas were lesioned, who didn't have functioning amygdalas, you know, was the blue square supposed to shock you? They'd say, yeah, that blue square, that's something I'm supposed to be afraid of. It's going to shock me. And so they kind of consciously knew that the blue square was something that they should be fearful of, but their body didn't really seem to be too worried about it, which is completely the opposite of people with hippocampus lesions. People with hippocampus lesions, you would expect them, they would show people with hippocampus lesions who don't have the ability to form long-term declarative memories. People with hippocampus lesions, if you ask them, is that blue square, is it something you should be fearful of? They'd be like, no, I'm not really fearful of that blue square. Why would I? Because they don't have a declarative memory to remember it with. They don't remember being told earlier that the blue square was something they should be afraid of. And so if you ask them, are you afraid of the blue square? They'll say, no, I'm not. These are the hippocampus lesions people. They'll say, no, I'm not afraid of it. But if you measure these individuals with hippocampus lesions, their physiological responses, in particular like their skin conductance, you would predict that they would show increased skin conductance response. Was what once again, this kind of shows this separation between implicit aspects of the mind and explicit aspects of the mind. People with amygdala lesions, they show the implicit response. Sorry, I need to correct myself. People with amygdala lesions do not show the implicit response. That is, they don't show changes on skin conductance, although explicitly they know I should be afraid of the blue square. People with hippocampus lesions, it's just the opposite. They do show implicit responses. They will show skin conductance increases when they see the blue square. But if you ask them, are you scared of that blue square? They'll say, no, I'm not. So the people with amygdala and hippocampus lesions kind of show opposite approaches and the way they deal with the emotion in this instructed fear paradigm. And this little statement right here, increases in skin conductance for the blue square and skin conductance correlated with amygdala activity. This is this figure right here. Any questions or comments about instructed fear paradigm, how amygdala lesion patients responded, and how hippocampus lesion patients responded? Okay. Okay, so another topic, arousal enhanced consolidation. This is a topic which is still involves the amygdala. At least the amygdala is theoretically the most important aspect of the neurocognitive mechanism underlying this. And I guess it's a good time to remind you, the amygdala is directly adjacent to the hippocampus. And the hippocampus we know is important for forming long-term declarative memories. And I guess the way I view the amygdala is that the amygdala can be involved. It is important for perceiving emotion, experiencing emotion, both cognitively and physiologically. But one of the functions of emotions is to change the way or the strength of a memory which is formed during the experience of that emotion. I guess a kind of related example of this would be flashbulb memories are kind of form of arousal enhanced consolidation. The go-to example for me would be 9/11, September 11th, 2001, in which case that day, yeah, it was a Tuesday. I had differential equations that day. And, it was really stressful that morning because seeing the planes slam into the towers. And I think I saw the towers fall right before I left to go to my differential equations class. And I actually remember being in differential equations class and just being kind of like not really able to pay attention. But I have a pretty clear memory of watching the professor's mouth move, but like nothing was really clicking in my head actually that day. But I still remember a lot of the elements of experiences of that day, where any other day from 2001, I don't actually remember that many things. But that particular morning, I remember many of the things that occurred to me that day. So I had a strong, stressful experience, which caused me to form declarative memories about episodic memories in particular about various things that I did that day. I remember also waking up that day and I remember hearing usually I woke up and there would be music on my radio, but this day it was news because of the September 11th thing. So there was aspects of the stressful experience which caused me to have long-term memories of that day that are still pretty strong even today. So that would be an example of 9/11 is an example of a flashbulb memory. And it's kind of like arousal enhanced consolidation in the sense that arousal enhanced consolidation is the situation in which when you're experiencing strong emotion, usually it's a stressful emotion, that this can actually improve learning. This can improve the rate or the effectiveness of memory consolidation. And so I have an example right here on the next slide. So in this study I'm about to tell you about, the stressful situation is in a laboratory, was people had to perform a cold presser task. So a cold presser task is when you have to dip your hand in cold water and you have to hold it there for a long time. Really, usually in the cold presser task, they just ask you, to hold your hand in the cold ice water for as long as you can take the pain. I mean, it's stressful because, any type of task where you're having to take the pain as long as you possibly can is a stressful thing. So that was kind of the stress-inducing thing in this particular study. Around the same time, so they would do the cold presser tests, and at the same time, they were looking at slides, pictures of objects on slides. And then the control condition would be people that didn't take the cold presser test, but they were still looking at the slides. And they wanted to see, how does this stressful situation, taking the cold presser test, how does it, or this cold presser experience, how does this influence your ability to remember the slides later on? So remembering the slides, this is the researchers trying to get you to form a long-term declarative memory of this thing that you've seen. Is the memory of these slides better or worse depending on whether you've performed the cold pressor task, which stresses you out, it is arousing, compared to a control condition in which you do something which is relatively less stressful, less arousing. And so I think I have this is the abstract. I'll come back to that slide in a second. This is the abstract, so you can read this on your own time. No, this is the one. This is the one about arousal enhanced consolidation with the cold pressor test right here. And basically, so if you want to see this, if you want to read more about what the study I'm talking about, this is the abstract, and it's condensed version of kind of what I'm saying right here. And basically what they suggested, there with a little bit of qualification, basically what they found is the individuals in the cold pressor condition did later remember the slides better than the individuals in the control condition. So this suggests that when you're in a stressful situation, like in this case, the cold pressor test, that it improves your ability to remember certain types of things. In this situation, it was slides. So emotion, stress in this case, can actually improve your consolidation, your memory of things that you're later tested on. Yeah, and the book also talks about the Morris water maze. So they use the Morris water maze as the example of arousal enhanced consolidation. So if you remember in the Morris water maze, well, I want to make sure that we still remember this well. What is rats in the Morris water maze? Does anyone remember what is the thing that they have to remember? If you are a rat in the Morris water maze and you're forming a declarative memory in the Morris water maze, what exactly is the declarative memory that you're forming in the Morris water maze? Anyone remember that?
Is it the platform that you get up on?
Yes, it is. Can you be a little bit more specific? What about the platform?
I just remember they're swimming around in like a tub of water, and they just always get dropped in at like different locations, and they have to get up onto a platform so they're not just swimming around in water.
Exactly. So the location of the platform is the thing that they have to remember in the Morris water maze example. In particular, they have to be able to use those landmarks on the wall to help them remember where the platform is. And the book suggested, although they don't talk a lot about it in detail, that rats perform better. Their memories of where the platform is better if after finding the platform, on like during the learning trials, when they're finding the platform, if they're given some type of stressful experience, some type of arousing experience. I believe the book, if I'm recalling, says this in particular, stressful, some type of a stressful thing like a loud noise or something that scares the rat. But potentially also it's not really clear if it has to be stressful, like negative things, or positive things. It seems like it can be either one. If I'm recalling correctly, the book said it was a negative experience. When the rat finds the platform during the learning phase, they give it some type of emotional arousing stimulus, and then that's the arousal part. And then they look later on, how are these rats performing when they're trying to find the platform again? And there's something about that arousing experience when you're in the learning phase and something stressful happens. In this case, the stressful thing happened right when the rats found the platform, the thing they were looking for. How does that affect their ability to later find the platform? And the book suggested that it helped consolidation. It made it so later on, experiencing that arousal during the learning phase caused you to have better performance in finding the platform later during a test phase. Questions or comments so far about arousal enhanced consolidation? Okay, so moving in a slightly different direction here. This is just a case study that's presented in the book. So we're moving in a different direction in the sense that we're now just talking about this case study instead of the amygdala as a whole. We're talking about what happens to an individual when their amygdala is damaged as far as the everyday experiences of emotion. And the main emotion, pretty clearly, so the amygdala is involved with lots of different emotional, subjective experience of various types of emotions, but the emotion that seems to be the most strongly impaired, if you have an amygdala damage like this patient, SM, the emotion which is most clearly impaired by amygdala damage is fear. So this is a production task where this participant, this this case study patient, SM, had to draw different emotional expressions on the face. And the patient could draw happiness, sadness, anger, surprise, and disgust, although that's not a particularly disgusted face, actually. But I would say most of these, the anger is pretty darn good, to be honest. I think it's better than I could do. Most of these emotions are pretty accurate. So you ask this patient, draw what these different facial expressions look like, the expressions of these different emotions physically on the face, what they look like. And the patient was pretty darn good for all of them, except for fear, afraid. And when asked to draw afraid, for whatever reason, the patient drew a baby. So this is part of the evidence that the amygdala is important for understanding fear. And I'm trying to think if I have the other. Here's the other. Here's some more evidence in animals when they're monkeys approach fearful objects. Yeah, so an animal with their amygdala damaged will have a previous experience with an object that's bad. That object shocked me in the past, or that object did something that caused me pain in the past. And so people with an amygdala which is intact, we learn from these types of experiences, okay, that's something that I should avoid. And so we're using our emotion of fear to help us avoid the thing that we have learned from previous experience causes us some type of pain, some type of negative outcome. But in the study, which is talked about in the book, when your amygdala is damaged, you'll still approach objects which cause you fear, even if your amygdala is damaged. And so fear seems to be the emotion which is most strongly tied to the amygdala. Individuals that, like this individual, SM, right here, not only could SM not draw fear, but SM had problems seeing fear in other individuals. Facial expressions of fear, they didn't register as being fearful inside SM as a result of the amygdala damage. So experiencing fear seems to be something which is impaired when you have amygdala damage, and then also perceiving fear out in the world. So kind of any aspect of your experience of fear, whether within yourself or seeing it in others, is reduced or impaired as a result of amygdala damage, whether in humans or animals like this monkey study right here. Questions or comments about the amygdala and its role in fear?
I know amygdala problems can cause like antisocial behavior. Did any of these studies suggest any antisocial behavior in these participants?
I actually didn't know that, and I don't know the answer off that. These are the ones that are in the textbook, and I don't remember if the textbook mentioned something about antisocial behavior. It did not commit to my long-term memory, and I don't actually remember if it mentioned that or not. And I haven't read these studies that I just talked about. I have not actually read these individual studies. I've read the textbook descriptions of these studies and more or less relaying what the textbook says about these studies. So I don't know if antisocial behavior is associated with amygdala damage, but I could see why it would be. So another cool thing about, here's another, so here's a study about, that kind of really emphasizes this difference between explicit and implicit aspects of emotion, which is really, in chapters 9 and 10, there's really a lot of emphasis on the difference between explicit and implicit. In chapter 9, it's the difference between explicit and implicit memory. Patient HM the hippocampus was removed, and although he could still form procedural memories, which are a type of implicit memory, his ability to form explicit memories was impaired. We've talked about these dissociations that occur in emotion as well, and here's another example of one. So this is an individual, this Edouard, I'm not even going to try and pronounce the last name, is a doctor. And the story, to my understanding, is he would have like a pen in his hand, and he would cause a patient, every time he would see the patient, the patient would shake the patient's hand with the pin. I also think it's kind of like, I remember my friend Matt Stutzman in the day had like a little shocker thing that you would shake his hand or whatever, and he would shock you a little bit. You can kind of think of it like this. It's basically a little, I shake the person's hand and I get a small painful experience. And this doctor, kind of a mean doctor, did this to a patient, and the patient had an intact amygdala, but the hippocampus was damaged. The hippocampus was damaged, but the amygdala was intact. And so if you ask this patient, if you ask this person with the damaged hippocampus, the amygdala is intact, but the hippocampus was damaged. If you ask this person, this doctor, have you met this doctor before? They're going to say, no, I haven't met this doctor before. If they ask you, has this doctor in the past done things to you which have annoyed you, poking you with a pin or shocking you? Those are things that would annoy you. Has he done things which has caused you pain? The patient can say, no, I've never met this person before. Because the patient, as a result of having no hippocampus, is not going to have the explicit memory. However, if you actually look at the patient's nonverbal behavior, after a while, even though the patient would say, I've never met this doctor before, as far as I know, this doctor is a nice person. But if you actually looked at the patient's nonverbal behavior, it was pretty clear that the patient, when seeing the doctor and then the doctor held out his hand to shake the hand, the patient hesitated. The patient was quite reluctant. It would take, would not readily reach out the hand like he or she would for most people, because the patient was kind of implicitly learning. The patient had learned, even though he didn't explicitly consciously remember, I've met this doctor before and he shocked me in the past, there was something in the patient's body where they kind of remembered implicitly, there's trouble here. So they weren't saying it in words, but their body still knew, if I shake this hand, something bad is going to happen to me. And the part of their brain that was allowing them to do this, to implicitly know I should be fearful of this, was the amygdala. The amygdala helps you form kind of implicit memories related to fear. And if the amygdala is intact, then your body still kind of knows I should be a little bit afraid of this thing, even if consciously, as in this person, because their hippocampus was impaired, even if consciously you don't realize that you should be fearful. It's really emphasizes the separation between explicit knowledge of emotions, explicit memory, versus the implicit memory, which is something that's like our body knows that our brain might not necessarily allow us to be consciously aware of. Any questions or comments about this? Okay, the last topic, I think this is the last one. Yeah, the HPA, we have two little topics left. The HPA axis, if we were in class, I would show you this video, but I'm just gonna have you watch this video on your own about the HPA axis. The HPA stands for hypothalamus, pituitary gland, adrenal glands. These three parts, the hypothalamus and the pituitary gland are part of the brain, obviously. And please note, the hypothalamus, which is this brain region we're talking about, this is still in the lower parts of the cortex. You get a nice view here. All these brain regions we're talking about in this whole section of emotion. We're talking about brain regions that are kind of in the lower half of the brain. We're not really talking about the frontal lobe that much. We're not really talking about the parietal lobe that much. We're talking about kind of these lower emotional, kind of these core emotional brain regions. And the HPA axis is kind of I think of it as like a loop where signals are sent from the hypothalamus, and the hypothalamus and the pituitary gland are working together, and they send signals down to the body. And so first off, I wanted to point out that we're talking, the emotions we've always talked about. There's the cognitive component, which is like, we think of that as the mental thing. But then there's also the physiological feelings of emotion. We're talking about like muscle tension, your heart beating, hormones are being released. And so these are things that are occurring in your body, and simultaneously we're having a mental experience, but we also have this physiological bodily experience. And the HPA axis really encapsulates kind of the two aspects of emotion here, in the sense that there's definitely these parts of the brain, the hypothalamus, pituitary gland, which basically stimulate the release of hormones. down here from the adrenal glands. These are the kidneys right here. And my understanding is that the adrenal glands are basically right on top of the kidneys. The adrenal glands will cause the release of a stress hormone in particular. Let me move my picture so you can see it. Cortisol, but also these neurotransmitters right here will be released into the body. So basically these are hormones, neurotransmitters that are released in the body as a result of some type of stressful event. The brain regions can stimulate the release of chemicals into your body, which will change the way that the body is going to interact with the environment. When you're stressed out, the body becomes stressed out through the release of these different hormones, which will cause you to, like fight or flight, for instance, something you're more likely to do when you have cortisol released into your body. So the brain is basically telling your body, okay, you need to be in stress mode. And it's a loop though, because then there's parts in the brain which are basically able to sample the blood. They're basically able to sample what's going on in your body right now. And when a certain amount of the hormone, when it can sample, okay, there's a lot of the stress hormone in the body right now, it can turn off. So there's this loop. The brain can cause the adrenal glands to release stress hormones. And then the stress hormones travel through the bloodstream, they travel through the body. And then eventually that information is reaching back up to the brain, the concentration of these stress hormones and neurotransmitters throughout the body. And then once a certain level or degree or concentration of these hormones has been reached, the hypothalamus can then signal, okay, it's time to turn off. We've had enough stress. It's time for this to be turned off. It's a response that makes a lot of sense. I need to get the body stressed, but once the body is stressed enough, it needs to chill. We need to not get too stressed out. We want it to be a productive amount of stress to help us deal with some type of stressful situation which is occurring out there in the world. So that's why I call it a loop. And there's some interesting things about the HPA axis and how it is influenced by development. And so I like the HPA axis because it kind of explains how some people respond better to stress than others. And part of the idea is that your early life experiences kind of influence the way your HPA axis works. And if you have really stressful experiences early on in life, it can kind of predispose you to overreacting to stress later in life. One potentially proposed mechanism is that if you have a lot of stress early in your life, it makes it so that shutoff switch doesn't work as well. And so you have a stressful experience. It's good that your body gets a little stressed out in a stressful experience because it can help you deal with the stressful thing in the environment. But for some individuals, it's hard to turn off the stress switch. And presumably, some evidence suggests that early life stress early on your life will make it so that later on in your life, the shutoff switch doesn't work as well. So people that have really strong stress early on in their life are going to have problems dealing with the stress later in life. There is some evidence for that, and it comes from this study. These are rats. And I should point out that they're all male rats. There actually is some evidence that the findings that we're talking about are significantly stronger in male rats than they are in female rats. And so these findings do not necessarily generalize to all rats. In this study, they only generalize to the studies generally are stronger for the male rats than they are for the female rats. But we'll talk about the findings. So here is a study where they're doing, I think does it talk about it here? Yes, a depressive-like behavior in the forced swimming test. So it's kind of a mean task, but basically what they do, they can decide whether an animal is depressed or not by the amount of time that it will try to escape a swimming pool that it's kind of stuck in. So they put it in like a Morris water maze type of situation. And rats do not like it. They want to escape at all costs. And rats differ depending on, they measure depressive behavior in this forced swimming task based on the rats that are depressed are the ones that when they're put in the water, instead of trying to escape instead of trying to swim around and find ways to get out of this, they kind of just sit there and they'll just do enough just to not drown. So they'll kind of float and then they'll start to sink a little bit and then they'll bring themselves up just so they don't drowned, and then they'll kind of, gradually sink down a little bit. And then once they get down and their nose is below the water and they start sinking, then they'll bring themselves up just enough to not drown. Those are the depressed rats. Compared to the rats that are not depressed, they'll constantly be trying, they'll be working, and they'll be attempting to productively escape from this stressful environment. And so in this study, what they do is they want to know, does early life stress, does it increase these rats' susceptibility to this depressive like behavior later on in their life? And so the way they stress out the rats when they're young is that it's called maternal separation, which is what MS stands for right here. So they stress out the rats early on in their life, not the entire time, but for certain segments of the day. They will remove rats when they're baby rats, pups, from their mother. And presumably this is a stressful thing for these baby rats. So they're used to being around their mother almost all the time, but for a couple hours every day for a certain period of time earlier on in these rat babies' development, they'll remove them from the presence of their mother. And this is stressful for them. And I'll present some evidence. This is some evidence, and I'll present some more evidence on subsequent slides. Compared to AFR, I don't actually remember what that exact acronym stands for, but that's definitely the control rats. These are rats that did not have maternal separation. Their childhood was not particularly stressful compared to the MS rats, maternally separated, whose childhoods were more stressful because of being separated from their mother periodically for several weeks when they were babies. And what they wanted to see, is there a difference between the maternally separated rats and the control rats on their depressive behavior in this forced swimming task? So this is the forced swimming task we're looking at here. Immobility, that means you're depressed. If you go into the swimming pool and you're a rat and you just don't try, if you're immobile, that means you just go there and you kind of sink down until your nose gets underwater, and then you just do enough to get your nose back above water again, and you're just barely staying afloat. That's being immobile. And what they find is the maternally separated rats are more immobile. So that would contrast with the control rats. So the white ones, they're less immobile. If you're less immobile, that means you're swimming around, you're trying to find some way of escaping. I think the analogy is, even though it's rats, and rats' sense of depression is clearly going to be, experienced different than humans, it's kind of a good analogy. It's kind of a good metaphor or comparison to humans. In humans, when a human is depressed, they are less likely to, want to be involved with activities that you usually would find joy in, or in this case, this would be an activity where you're trying to, save your life, get into a safer situation. And the depressed rats are less. Rats are less willing to exert effort to try and get to a better situation. Questions or comments about the forced swimming test and how maternally separated rats differ from controls in the forced swimming test? So here's another study that's on maternally separated rats. This one's using the Morris water maze in the more classic sense of the Morris water maze. This is not forced swimming anymore. This is like the classic Morris water maze test. And so for this, they have a group of maternally separated rats. When they were babies, they were separated from our moms, compared to the AFR, which is the controls. And then they have them learn the Morris water maze. And we just talked about it earlier in this class session. The thing that you're supposed to learn in the Morris water maze is the location of the platform. And you have those landmarks on the wall, which helps you remember where the location of the platform is. And you're having to form a long-term declarative memory of where the platform is relative to those landmarks. In this study, they divided up the Morris water maze. It's a circle, but they divided it up into 4 quadrants. In case you're curious, the quadrant 3 was where the platform was. So what they do is they give training. They have them maternally separated in the control rats, and then they give them training in the Morris water maze, where they basically, over a series of days, once they're adults, they're no longer, they were childhood when they were separated from their moms. But then they're later on, this is when they're adult rats, then they have a learning phase where they're trying to learn, where is the platform? You know, they have to use those landmarks to remember where the platform is. They give them several days of experience with the Morris Water Maze so that they can find out where the platform is relative to those landmarks on the wall. The platform really during these trials was during these learning phases it was in quadrant 3, Q3 there. Then during the test phase, which is what you're seeing here, they're measuring the amount of distance swimming in each of the 4 quadrants. They probably could have also used the amount of time in each quadrant, but basically there's a test phase, and during the test phase they're a little bit mean. They remove the platform completely, and what they want to see is what they assume is the rats that remember where the platform is. They're going to spend a lot of time swimming where the platform used to be. So time is not actually the dependent variable though. They do the distance move. So this could be the amount of distance within each quadrant that the animal is swimming. So you can imagine the animal's like, okay, I know the platform's around here somewhere, so I'm going to swim around this location right here until I find the platform. The platform's not there though, because they're kind of mean during the test phase, and they completely remove the platform because they really just want to see, are the rats able to figure out where the platform is supposed to be? And they compare the control rats to the maternally separated rats, basically, for the amount of time or the amount of distance swum in each of the quadrants. The rats should spend a lot of time swimming in Q3, quadrant 3, but you actually find that the control rats spend significantly more time swimming in Q3 than the maternally separated rats. So what this suggests is that not only does this early life stress of being taken away from your mother, it might be like a predisposition to depression, but it also influences your ability to form long-term memories later in life. This is a cognitive thing. This is a cognitive task, your ability to remember where the platform is relative to these locations on the wall. The control rats, they showed evidence that they remembered quite strongly, yeah, the platform is in quadrant 3. And you know that because these rats, they moved around a lot in quadrant 3. They moved around significantly more than the maternally separated rats. The maternally separated rats, for whatever reason, spent a lot of time swimming around in quadrant 1. They spent more time swimming around in quadrant 1, even though the platform had never been there. Compared to quadrant 3, they actually spent more time swimming around in quadrant 1, even though there was no reason to be, than they did in quadrant 3. So what this shows is that early life stress can influence your future cognitive abilities, at least in rats. If you have early life stress, maternal separation in this situation, it's going to influence your ability to solve, cognitive problems, finding the location of a platform based on these landmarks on the wall. Any questions or comments about this? And so here's evidence that, I presented these findings right here. I guess I should go back. This finding right here, this is a purely behavioral finding. They didn't, in this study I'm not talking about animals that have their brain lesions. This is comparing stressed out rats to non-stressed out rats in a behavioral response, how much time they swim trying to escape from a pool that they want to escape from. And the maternally separated rats were more depressed. It's a behavioral response. This one right here, this is behavioral as well. It's a cognitive task, learning where the platform is relative to the locations on the wall, but we haven't lesioned any brain regions. We're not talking about anything physiological yet. So you might be wondering, what does this even have to do with the HPA axis? Some of the evidence from the same study does suggest that these behavioral findings that we were just talking about are related to the development of this HPA axis. Remember, the H stands for hypothalamus, and it's that loop where the hypothalamus, via the pituitary gland, will send signals down to the adrenal glands, which will cause stress hormones to be sent. Those stress hormones kind of find their way back up to the brain, where the brain can then stop the stress response, which can have various types of emotional and cognitive effects. So this study right here, this aspect of the same, these are all part of the same study, shows that maternally separated rats have much higher levels of cortisol in their bodies in response to a given stressor. So they don't say what the stressor is, but they say cortisol and another related stress hormone right here These are basically both stress hormones. And they're comparing the amount of the stress hormone for the control rats compared to the maternally separated rats, the control rats compared to the maternally separated rats. And it says here in response to an acute stressor. Oh, and they do say it's 15 minutes of swimming. So that's the stressor. The stressor was being forced to swim for 15 minutes. And they wanted to see what is going on physiologically inside the bodies of these rats. And so they measured these stress hormones, and they do find the maternally separated rats have higher levels of the stress hormone in response to the stressor compared to the control rats right here. And so this appears to be that phenomenon I was talking about earlier on. When you're early on in life, for this feedback loop, the HPA axis is kind of learning. It's acquiring properties based on its environmental experience, your environmental experience. And one of the things that appears to happen in individuals, at least these rats that have early life stress, is that the feedback loop becomes less efficient. Eventually, the hypothalamus should be getting signals saying, you have a lot of this stress hormone in your body right now. We need to stop the production of cortisol so that you can reach a more productive level of cortisol. There's too much cortisol right now. We need to chill it out. But you can see the rats with the maternally separated rats, for whatever reason, their HPA axis is not able to apply the shutoff switch. And so the cortisol levels are ramped up. The stress hormone remains high in these rats who earlier in their lives had experienced this maternal separation. And so this is an ongoing field of research. It's not exactly clear how these elevated stress levels are causing all of the other behaviors that we talked about. For instance, the maternally separated rats, although they have higher levels of cortisol, a stress hormone, they'll actually try less hard to escape. So they have high levels of stress, and it's almost like they're so stressed that they won't even try to escape when they're in the swimming pool, the forced swimming test. And their cortisol levels are very high, and maybe that's the reason they have some difficulty concentrating when they're in the Morris water maze, and they're having trouble remembering where was the platform relative to these locations again. And this is an ongoing field of research. How are these high levels of cortisol? How are they influencing other behaviors? And in general, the hypothesis is that maternally separated rats have problems with the regulation of this HPA loop, where they can't turn off the cortisol. Questions or comments about the HPA axis? That's all I have about that. So yeah, these three rat findings are all from a highly cited study on rats and the HPA axis. The last thing I want to talk about is the insula. I don't have tons and tons to talk about tons and tons to say about the insula. It's relatively higher up in the brain. So we've been talking about like the amygdala, which is down here, and the hypothalamus, which is also relatively lower. So we're moving up, when we're talking about the insula, we're moving up closer to the frontal lobe. It says here the insular cortex is tucked between the frontal and temporal lobes in the Sylvian fissure. So the Sylvian fissure is right here, also known as the lateral fissure is right here. If you open that up and then you go towards the middle parts of the brain, that's where the insula is. So it's definitely tucked away inside the brain. It's lower than the frontal lobe, but it's higher than the amygdala. So amygdala is down here. The insula is a little bit above it. So it's closer to the frontal lobe than the amygdala is. So it's probably an evolutionarily more recent emotion region. And it talks here about some of the connections. The insula does have connections with the amygdala, but it also has connections with the prefrontal cortex. So this is a region, you can kind of think of it, maybe the insula as kind of a bridge between some of those core emotion regions like the amygdala, which are low in the brain, and some of the higher regions like the prefrontal cortex. The prefrontal cortex is like this stuff over here. The insula is kind of like a bridge that allows signals potentially to be sent back and forth between the frontal lobe and like the amygdala and some of the other lower emotion regions, which are down in the bottom parts of the brain. The insula seems to be kind of an integrative or allowing communications between frontal lobe regions, which are known to be important with like cognitive memory types of things, and the more core emotion regions, which are down below. So here's some kind of a highlight of some of the interesting findings about the insula. We're not going to go into tons of detail about the insula. But probably one of the most interesting aspects of the insula is that activity in the insula you can see activity in insula when people are experiencing like emotional issues in various emotional states, but also physical states. Um, so yeah, the example here is abdominal cramps. And so experiencing kind of physical pain is associated with activity in the insula, similarly to the way that it's experiencing emotional pain, purely cognitive, mental pain. So this is an interesting thing because it suggests that the insula is part of the brain which might kind of bridge together physical states and mental states. We know that emotions are subjective, cognitive, mental things, but they're also physical things. They're both sides of the equation. And the insula seems to be a region of the brain which might be kind of integrating purely mental aspects with physical aspects of the emotional experience. Because the unique property is the insula is activated. It's associated with physical experiences in a way that the amygdala and some of the other brain regions they're not necessarily activated in response to like physical pain, like the insula is. Some also cool evidence, which is kind of along the same lines, is that the insula activity correlates with interoception, which is a very interesting phenomenon. Interoception, I think I defined that in the notes portion of the slides. Interoception is basically your ability to detect, to feel internal states of your body. So like some examples, probably it's kind of, it's a little bit weird, but probably one of the best examples is when you sense that you have to go to the bathroom. You have basically something going on inside your body and you can sense that. You're feeling something which is going on inside your body. But I spelled it wrong, but this is supposed to be heartbeat. You actually can sometimes hear your heartbeat. Thirst is another example. Feeling like abdominal things, your abdominal area is one of the areas that when you are feeling stressed, that's one area that people are likely to say they report feeling some type of physical sensation. You're having a mental experience of stress, but you're often likely to feel some type of physical issue down in your abdominal area. These are examples of interoception, feeling that stuff down there. There's something inside of you which is going on and you can feel it. We're used to thinking about our sense of touch as being like on the outside of our body. When we're talking about interoception, we're talking about feeling things which are inside of our body. And there's some evidence that the insula is important for this ability. Not everyone is as good or as in tune with their internal bodily states. And the extent to which your insula becomes activated when you're performing interoception tasks, when you're trying to feel your own the internal states of your body, the insula seems to be important for that ability. And that's really, it says here, lesions to insula reduce self-reported arousal in response to picture stimuli. So what this shows is that when you have insula lesions, we know it's important for all these physical things up here, like interoception, the experience of abdominal cramps, things like that. But there's also clearly emotional impairments that occur when your insula is damaged. Like this one's pretty striking, the inability to see to tell the difference between positive and negative emotional pictures. So it's almost as though, we know it's important, the insula is important for physical things, but it's also important for seeing a person out in the world and being able to read their emotional expression. Is this a positive experience this individual is expressing, or is it a negative expression? And so what this really supports the idea that the insula is important for kind of mental aspects of emotion, but physical aspects as well. And all this kind of together, which is the point I've basically been making, insula might integrate cognitive, and by affective, I mean bodily aspects of emotion. So that is all I have for this chapter. We have a little bit on chapter 11, which we'll do Wednesday. We'll also be doing a study guide for the last test on Wednesday as well. That's it. We have one more class and then we're done. And as I mentioned at the beginning of class, if you do want to, I will allow the final in-class lecture quiz to be optional. Basically, it'll be extra credit. You basically have to supply the topic, and then I'll give you a question that you have to answer on that topic. Some topic that you want a little bit of practice with. You'd say, this is a topic I'd like to practice with, or this is something that I'd like to know more about. And I will give you a question that you answer. You'll turn it in via Blackboard. I'll call it something like extra credit, lecture quiz, extra credit in class assignment, something like that. And you turn it in, I'll give you a couple points extra credit. So you guys can take off if you want. I'll stick around until everyone's gone if people have questions. Otherwise, feel free to email me if you want to make Zoom meetings or have some type of meeting.